In this segment, I'm going to talk about sensory integration and neural circuits. We're going to draw a neural circuit to cause a rapid escape reaction, retraction, in an adenoid worm only to the combination of 50 Hz vibration, an odor from a predator, and daytime. Whenever you're doing these, you need to break it down into pieces, detect each of the individual sensory inputs. Those get integrated in one or more interneurons, which then affect motor neurons and cause changes in muscles in sequence. So in this problem, we're not going to show all the details of how each receptor works because all we need is a neural circuit. We'll start with a sensory hair, and we'll specify that this hair is sensitive to vibration, to sound waves. It could be coming in water, vibration in water or air, and it makes the hair move back and forth, and that frequency of movement activates this neuron A that has sodium stretch channels. So that's A. B, an odor from a predator binds to a G protein coupled receptor in a cell, and through mechanisms we've talked about results in neurotransmitter release. And C, light, which is another G protein coupled receptor. The receptor is rhodopsin, and so light activates this receptor. Again, we get neurotransmitter release, and so in neurons A, B, and C, we have neurotransmitter release that's causing action potentials. We need to integrate these at some cell D that will produce action potentials only when all three of these inputs are present. So if we have 3 and we need to depolarize to -45, we could have each one depolarize plus 10 millivolts. So each at plus 10 millivolts, and that'll get action potentials in interneuron D. For the escape reaction, in an annelid worm, we have longitudinal muscle cells, and we need to contract the longitudinal muscles in order to get an action potential. So motor neuron E will cause action potentials that release neurotransmitter to the muscles and cause retraction. Now we check to make sure it works. We have vibration that causes that hair to bend back and forth, stretch channels within it, and this cell will produce action potentials only if there are 50 of them per second. The odor from a predator activates a G protein coupled receptor. I haven't shown any of it, but it releases neurotransmitter to B and causes action potentials in B. And rhodopsin is absorbing photons, releasing neurotransmitter to C. These are integrated at the interneuron because each depolarizes that interneuron only by 10 millivolts, which add together to depolarization of 30 millivolts, taking the cell from -70 to -40, which will cause action potentials. That releases neurotransmitter at E, which produces action potentials that cause contraction of these muscle cells. What would we do if we wanted to make this a neuroendocrine system? So we wanted to include hormones. In this case, the question is, we want to prevent the escape reaction if the worm is reproductively mature. So in other words, if there's some hormone, let's call it reproductive hormone. Here's the circuit that we had before. We have vibration that's moving that. We have an action potential in A only if there are 50 action potentials per second from the sensory neuron S. The predator compound binding to a G protein coupled receptor causes neurotransmitter release to B, and photons, light, to C. So each does a 10 millivolt depolarization that gets us to -40. We have action potentials in D, contraction of the muscles. So what we want to add is block this in the presence of reproductive hormone. We can predict then that what we want might be inhibition that might block it by taking us -30 millivolts. If cell F is producing action potentials, it'll open potassium channels that will depolarize this -30 millivolts. So reproductive hormone has to affect a cell it could bind, if it's lipid insoluble, to the surface and activate a second messenger pathway to release neurotransmitter to F, and that opens sodium channels, depolarizes F, and causes action potentials to block it. So now if we have vibration, predator, photon, in the absence of reproductive hormone, we get action potentials in D in the escape reaction. But if this worm is reproductive, then all these things happen. We get 10 millivolts depolarization from each, 30 total, but at the same time, reproductive hormone is causing action potentials in F, which have a minus 30 millivolts, blocking the effect of these others. D fails to fire action potentials, and there's no escape reaction.